Hello, welcome back to microbiology. So, in this clip, we are going to talk about some of the significant historical things that have happened within the microbiology world. So, first big history thing was when life started. Obviously, that's kind of important. So, the bacteria we have now, they are not the actual bacteria that were the first life on Earth, but their ancestors were the first life on Earth. For much of the history of our Earth having living things on it, those living things were prokaryotes. But as people came along, for much of the history of humankind, we had no idea that those prokaryotes existed. We have had no idea that anything microscopic existed before we started having microscopes. So in 1665, Robert Hooke, who is this guy, and again, I'm not going to ask you to memorize dates, but these names you should be familiar with. If I ask you, what did this person do, you should be able to tell me what that person did. And if I ask you who did this thing, you should be able to tell me what that person's name is. Okay, so let's start with Robert Hooke. He was looking at little slices of cork under a microscope. So cork is a type of plant, and he noticed they look like little squares. They look like they reminded him of the little cells that the monks lived in. And so he said he named them cells. They look like little cells. Today we would say he was probably looking at their cell walls. If you look at a plant cell under just simple magnification, you can see a plant cell wall. So that was kind of a very important step towards understanding what cells are and what life is made of. In 1673, the first microbes were observed. We'll talk about that in a little bit. In 1858, Rudolf Virchow, who is this guy, he said that cells arise from pre-existing cells. So at that time, it was pretty obvious. Elephants come from elephants. People come from people. But lower life forms, there was still some debate. You know, it kind of looked like frogs might just be born out of mud. Worms just naturally arise from rotting meat. And bacteria probably just naturally arises from the air, maybe dirty air or something like that. But Rudolf Virchow said, no, cells come from pre-existing cells. That fly that's coming from your meat is actually probably some eggs from another fly. It had a mother fly that laid eggs that it was born from. That bacteria that's growing, it probably has a mother cell, or it definitely has a mother cell. All cells come from pre-existing cells. Today, neither of these statements are very fanatical. They're both pretty obvious to us to think about it. But at that time, it was not necessarily, like, it took a while for these statements to be accepted. But together, saying that all living things are made of cells and that cells come from pre-existing cells, these two statements combine to create what is cell theory. So if I ask you what is cell theory, I am asking you to tell me those two statements, that all living things are composed of cells and cells come from pre-existing cells. Now, if you're using a different textbook, it might have a couple of other statements combined in with what cell theory is. And they're all perfectly good statements too. But we are just going to stick with these two. All living things are made of cells, and cells come from pre-existing cells. Okay, so let's go back to Anton van Leeuwenhoek. So here we said, when are these first microbes observed? That's what Anton van Leeuwenhoek does. So Van Leeuwenhoek or Leeuwenhoek, or there's lots of different ways you can see it pronounced. I don't have a great Dutch pronunciation, but he was a Dutch merchant by trade. He was not professionally a scientist. He only did it kind of on the side as a hobby, but he started making microscopes. He just loved looking at things under microscopes. He would look at anything he could get his hand on. He would look at rainwater, feces, teeth. He would look at sperm. The first thing he looked at was ground up flakes of pepper. Anything he could get his hands on, he would look at in the microscopes. And in a lot of these samples, he saw living things, kind of squiggly little things moving around, and he called them animalcules. He drew them. He sent them to the Royal Society of London, where you could still see them today if you went and visited. And Robert Hooke repeated some of his experiments and confirmed his findings, these little animalcules. It's very likely that a lot of what he was looking at were probably amoebas. He may have been looking at other unicellular protists. He possibly also saw some larger bacteria in his cells, but he was the first person who actually saw microbes and sketched them and published his findings. So there was this ongoing debate at the time. One idea was spontaneous generation. Spontaneous generation is this hypothesis that living things come from non-living matter. So that frog from the mud, that maggot from the rotting meat, probably there was just some vital force in the air that formed life. And life just came from that force, from the air, just this life-giving ability of the air. Kind of related to this is miasma theory. Miasma theory says that diseases, this could be anything from cholera or chlamydia or the Black Death or plague, were caused by miasma, which is a way to say pollution. So bad air causes miasma. Scientifically, if you think about it, it's not a bad idea, not a bad theory, because a lot of things that smell bad, a lot of things that are polluted, have a lot of germs in them, right? So fecal matter makes the air smell really bad. A lot of ways that bacteria can be spread are through things that smell bad. So I can't blame the scientists at the time for thinking that the bad air was causing it, even though today we know that's not true. An opposing theory was biogenesis. Biogenesis was the hypothesis that everything has a parent. So if we go back to Rudolf Virchow in 1858, remember, he was saying that all cells come from pre-existing cells. So this is biogenesis. So there was a lot of debate that went on between what is really happening. Is it spontaneous generation? Is it some form of spontaneous generation? Or is it really biogenesis? So 1668, and I want you to pay attention. I'm not going to ask you the dates, but just the spread of the dates. We were starting in the 1600s. Francesco Redi had 6 jars that he filled with meat and left the meat to decay. 3 of the jars he covered with a net, and 3 of the jars were uncovered, so flies were able to get to it. So those jars were covered with a fine net, no flies could get to, no maggots appeared, and the jars that were uncovered, maggots appeared. So I want you to think about it. Where did the maggots come from? What was the purpose of covered jars? And did this support spontaneous generation or biogenesis? If you can't really figure out the answers to those, please feel free to come talk to me. But I want you to be able to just stop and think about that for a minute. 1745. So we are now in the 1700s, the next century. John Needham had boiled nutrient broth. This is something that's great to grow just a wide variety of microbes on. You can grow fungi, things like molds on it. You can grow bacterial cultures. You can grow all kinds of stuff on it. So he took this broth. He broiled the broth. He boiled the broth. So there was no microbes in that broth, okay? So the broth itself was sterile. After he did that, then he put them in some flasks that he had, and he saw that there was microbial growth. So where did the microbes come from? He said this supported spontaneous generation. I would say this is an example of bad science. So I want you to stop and think about it. Why would he say this supports spontaneous generation? And why would I say that that's bad science? Again, I want you to be able to answer these questions, but if you can't, feel free to come talk to me about it. Lazarus Spallanzani, around the same time, he repeated this experiment, but he did it a little bit differently. He took broth, he put it in a flask, he sealed the flask, then he heated the whole thing. So he is sterilizing the broth and the flask. And when you sterilize both the broth and the flask, no microbes grow. I would say this is a lot better experiment than the one we just saw, where Needham didn't clean his flasks. He didn't sterilize his flasks. Spallanzani did. And that's why he saw no microbial growth. Does this support spontaneous generation or biogenesis? Then in the 1800s, so this is 200 years after we start saying that biogenesis is existing, Louis Pasteur is going to do an experiment to show that microbes are coming from the air. So he took nutrient broth, he put it in a flask, and then he heated it. When the flask was not sealed, there was microbial growth, but when the flask was sealed, there was microbial growth. So he said, this is showing my biogenesis. Microbes are floating around in the air, are getting to the contents of the flask and growing. But proponents of spontaneous generation said, no, it's not microbes in the air. It's the air itself, the vital force in the air. So this still didn't put an end to the debate. So he did one more, just, I think, absolutely genius experiment. He created these S-shaped flasks. So in the S-shaped flasks, he pours his nutrient broth in, and then he bends the flask into an S. And this S-shaped flask, the air can still get in. The air just travels through all of this. But the microbes get stuck here at the bend. The microbes don't make their way in. So he heats this whole thing, sterilizes the whole thing. Air is still able to get in. Microbes are not able to get in. And nothing grew. So he had this sterilized, even today, you could go see these on display. They're still sterilized. Bacteria is not growing in them. Finally put a rest to just the air itself won't contaminate. It doesn't have anything contaminatory about it. There's nothing in the air that's able to grow microbes. It is the microbes themselves that are in the air. Finally put an end to this debate and settled the science that biogenesis is indeed what is happening. This started the golden age of microbiology once we realized there are microbes in the air and they are growing things and doing things and they could be affecting your health. It started with Pasteur doing work on fermentation and pasteurization. We also learned a lot about the relationship that microbes have to causing disease, how we develop immunity, and how antimicrobial drugs can be created. So let's start with fermentation. So fermentation is something that yeast naturally do when they eat sugar and oxygen is not available. They take that sugar and they convert it to alcohol as a waste product. This is kind of popular among a lot of people because this is how beer and wine and other alcoholic beverages are made. However, there are also bacteria that will take that alcohol, digest it, and create acetic acid. That's also known as vinegar. People do not like drinking vinegar as much as they like drinking wine. So there were these wine bibbers that were making wine, and they wanted to be able to sell their wine before it turned to vinegar, before it was spoiled. And so Pasteur discovered that you can do something called fermentation. In fermentation, you're going to mildly heat your alcohol. It's a mild enough heating that the alcohol does not evaporate, but it's strong enough to kill those bacteria that are still in it that would convert the wine into vinegar. So he kills the spoilage microbes without evaporating the alcohol. So now that alcohol is able to, travel a greater distance and the merchants can sell it to people and make more money. This process of pasteurization is still used today in many alcoholic beverages, also beverages like milk. Milk is typically pasteurized before it's sold in the United States. So some other stories. Ignaz Semmelweis is one of my favorites. If I was going to marry a dead person, it would be Ignaz Semmelweis. I just love him. So Ignaz Semmelweis was living in Austria in the mid-1800s, and there was an outbreak of puerperal fever. Puerperal fever is an infection caused by a streptococcal bacteria that can get into a woman's birth canal, and it kills her. And so Ignaz Semmelweis was noticing that these women, when they were going to give birth, some of them, if they were giving birth in the morning, they were sent over to the side of the hospital where they were attended to with midwives. And the midwives were not having such a problem with puerperal fever. But the women who gave birth in the afternoon, they were sent to the other side of the hospital where they were giving birth attended to by doctors. The doctors spent their mornings down in the morgue examining the patients who had died of puerperal fever, and then they came upstairs in the afternoon and attended to the women who were giving birth in the afternoon. And so Semmelweis started researching what were the differences between these. What were the midwives or the doctors doing differently to have different outcomes? He noticed at the same time, women who were giving birth like on their farms at home were still just fine. They weren't having outbreaks of purple fever. And so he started thinking, well, these doctors in the morning, they're down in the morgue, all up in the bodies of women who have just died of puerperal fever. And then they come upstairs, and they're very proud of being a doctor. It was quite a sign of prestige. And so all of the blood and guts and stains all over their lab coat, all over their arms and hands, because that was a sign of being special and important enough to be a doctor. Ignaz Semmelweis had this wild fanatical idea that maybe, just maybe, after the doctors are dealing with the corpses, maybe they should just stop and wash their hands. Just wash your hand before you get elbow deep in the next woman who's giving birth. Some doctors kind of accepted the idea. The idea at least spread and became well known, but it wasn't very well received. He ended up dying in an asylum. He went crazy and died in an asylum. However, his work was spread around enough that Joseph Lister became familiar with it. And Joseph Lister kind of built on this idea and saying that not only should we wash our hands before we're doing something, but we should also do something to disinfect our patients before we're doing surgery. And so he developed this chemical phenol, which is a rather strong disinfectant. It's a type of alcohol. And he used that to clean the skin around where a surgery was going to happen. And that prevented surgical wound infections. This is why he is considered the pioneer of aseptic techniques. A lot of these aseptic techniques being things that we learn in lab. Lister also what Listerine is named after, which is another kind of disinfectant used to kill bacteria in your oral cavity. And then Robert Koch. Robert Koch's pretty important. We're going to come back and talk about him quite a bit when we discuss chapter 14. Robert Koch was trying to figure out what caused different diseases. He thought probably there were different bacteria that caused different diseases, but he really wanted to make sure. So he provided a series of steps. These are called Koch's postulates. You can go ahead and look that up if you want to, but we'll come back and talk about it in a couple of months, or I guess in about a month. And Koch's postulates are just a set of steps, a set of experimental steps that if you go through this whole process, you can prove that this first bacteria caused a specific disease. He was able to show what bacteria was causing anthrax. That was the first one he did. He also showed what bacteria were causing tuberculosis. These postulates have been used to show what is a causative agent of a lot of different diseases. Our next person is Edward Jenner. Edward Jenner is a guy that if he had been my neighbor, he would have been in so much trouble. He's very lucky he lived when he did and not in modern times with modern ethics considerations. So Edward Jenner was living during a time of smallpox outbreak. Smallpox is very deadly. It kills about half of the people it infects. So it was pretty terrifying to be living during an outbreak of smallpox. But he noticed that a lot of the milkmaids were not getting infected and were not dying from smallpox. And so he started talking to them, and the milkmaids were all like, well, yeah, we've all had cowpox. From milking the cows, we've got cowpox, and anyone who gets cowpox doesn't get smallpox. So he thought this through and thought, I am going to do something. He took some cowpox scrapings. He found someone with cowpox and took some of their pox pustules, scraped it up, grabbed his neighbor's 8-year-old kid, and scraped that cowpox scraping right into the kid's arm. The kid was exposed to smallpox and did not develop smallpox. Lucky for him. The kid was safe. And so he was able to say something about this cowpox virus is stimulating your body to protect yourself from smallpox. This was the beginning of vaccination. And this idea that you can be immune to something. Immunity is being protected from a disease. In his case, immunity is acquired by vaccination, although you can also develop immunity if you acquire and survive an illness. People who study immunity are called immunologists. And today we have lots of vaccines against a lot of diseases that protect us from getting sick. A vaccine is a culture of avirulent microorganisms. So stop and think about that. Culture means it's a group of cells, but they are avirulent. They are not dangerous. So we're using cells that are not dangerous for preventive inoculation. That means we are injecting it to prevent disease. So we are using something that is not dangerous to prevent a disease that could kill you. We're going to come back and talk about interferons later. Let's just mark that out for now. So at this point, you should be able to tell me, what is cell theory? What evidence supported spontaneous generation? And how was it eventually disproved? What is the germ theory of disease? What is the importance of Koch's postulates? What is the significance of Jenner's discovery? Make sure you can stop and answer those questions. Make sure you're comfortable with each of those people we talked about, who they were, and, in a sentence or two, describe what they did. Okay, I will see you back for our next lecture, where we're going to talk about some more modern things that are going on in biology. See you later. Bye.